Dear students, the topic is plant growth regulators or phytohormones or plant growth hormones. The growth rate of plants is controlled by a number of chemical compounds produced within the plants. These are called plant growth regulators or phytohormones or plant hormones. These phytohormones are organic substances which are synthesized in very small amount in one part of the plant and transport to other parts where they show their effects. These hormonal substances belong to five classes of compounds that you know: auxins, gibberellins, cytokinins, abscisic acid and ethylene. So the plant growth regulators are simple molecules of diverse chemical composition. They are indole compounds, adenine derivatives, carotenoid, terpenes and gases. Based on their functions, plant growth hormones come under two categories. They are growth promoting and growth inhibiting. Growth promoting are auxin, gibberellin and cytokinins, and growth inhibiting are abscisic acid and ethylene. Plant growth hormones are classified into natural phytohormones and synthetic phytohormones. Natural phytohormones come under two categories: plant growth promoters and growth inhibitors. Example for plant growth promoters are auxin, gibberellin and cytokinin. Growth inhibitors are ethylene and abscisic acid. Synthetic plant growth regulators are naphthalene acetic acid 2,4-D 2,4,5-T. The first plant growth regulator is auxin. Auxin was first discovered by Charles Darwin and his son Francis Darwin. They observed coleoptiles of canary grass which respond to phototropism. After a series of experiments, it was concluded that tip of coleoptiles was site of transmittable influence that caused the bending of the entire coleoptile. FW went isolated auxin from the tip of coleoptiles of oat seedling. Have a look on the physiological effect of auxins. Auxins were first isolated from human urine. The term auxin is applied to indole 3 acetic acid, in short IAA. Auxins are produced by growing apices of stems and roots, and they migrate from there to the regions of their action. This figure shows the action of auxin. Auxin spread equally down both sides of the plant. Auxin collect on the shady side. There are two types of auxin: natural auxin and synthetic auxins. Natural auxins are indole acetic acid and indole butyric acid. Synthetic compounds which function as auxin are synthetic auxins. Example: naphthalene acetic acid or NAA and 2,4-D 2,4-dichlorophenolic acetic acid. Have a look on the functions of auxins. Auxin initiate rooting in stem cuttings, promote flowering, example uniform flowering in pineapples, prevent fruit and leaf drop at early stages, promote abscission of older mature leaves and fruits, apical dominance. The growing apical bud inhibit the growth of lateral buds. Auxins induce parthenocarpy, example tomatoes, widely used as herbicides. 2,4-D is used to kill dicot weeds and does not affect monocot plant. So it is used to prepare weed-free lawns, controls xylem differentiation and helps in cell division. Next is the gibberellins. The pathogen for Bacani or foolish seedling disease in rice seedling was first identified as a fungus called Gibberella fugicori. E. Kurosawa reported the appearance of symptoms of the disease in uninfected rice seedling when they were treated with the sterile filtrate of the fungus. The active substances which are responsible for this disease was later identified as gibberellic acid or GA. More than 100 gibberellins were reported from fungi and higher plants. They are denoted as GA1, GA2, GA3, etc. Gibberellic acid was the first gibberellin discovered. Next we can see the functions of gibberellins. Here is the functions of gibberellin. Increase in length of axis in grape stalk, elongation of fruit and improve its shape. Example apple, delay senescence. The fruits can be left on the tree longer so as to extend the market period. GA3 is used to speed up malting process in brewing industry, increase the length of the stem in sugarcane which help to increase the yield, hastens the maturity period, thus leading to early seed production, promotes bolting. What is bolting? Internode elongation just before flowering in beet, cabbages and many plants with rosette habit. Next is cytokinins. Skong and his co-workers observed callus. You know what is callus? It's a mass of undifferentiated cells. Formation from tobacco stems when the nutrient medium with auxin was supplemented with extracts of vascular tissue, yeast extract, coconut milk or DNA. Skong and Miller later identified and crystallized cytokinesis promoting active substance termed kinetin. Cytokinins were discovered as kinetin, that modified form of adenine from autoclaved herring sperm DNA. Kinetin does not occur naturally in plant. Zeatin is a cytokinin isolated from corn kernels and coconut milk. Natural cytokinins are synthesized in regions where rapid cell division occurs. Example, root apices, developing shoot but young fruits, etc. Have a look on functions of cytokinins. Functions of cytokinins help to produce leaves and chloroplast in leaves, lateral shoot growth and adventitious shoot formation, that means counteraction of apical dominance, promote nutrient mobilization which help in the delay of leaf senescence. More functions are given in this flow chart. Next is ethylene. Ethylene is a gaseous plant growth regulator produced in large amount by tissues undergoing senescence and ripening fruits. Cousins confirm the release of a volatile substance from ripened oranges that hasten the ripening of storage unripe bananas. Later, this volatile substance was identified as ethylene, a gaseous hormone. Functions of ethylene: gaseous in form and rapidly diffusing. Gas produced by one plant will affect nearby plants. Fruit ripening. Epinasty, downward curvature of leaves, encourages senescence and abscission. Initiation of stem elongation and bud development. Flowering. Ethylene inhibits flowering in most species but promotes it in a few plants such as pineapple, bromeliads and mango. Sex expression: cucumber buds treated with ethylene become carpellate, that means female flowers, whereas those treated with gibberellins become staminate or male flowers. Have you heard of ethiphone? The most widely used compound as a source of ethylene. It is an aqueous solution and is readily absorbed within the plant and releases ethylene. It speeds up fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits like cotton, cherry and walnut. Next is abscisic acid. During mid-1960s, three independent researchers purified and studied the chemical characterization of three different kinds of plant growth inhibitors. Inhibitor B, abscission 2 and dormin. Later, all the three were proved to be chemically identical and named ABA or abscisic acid. Abscisic acid is a general plant growth inhibitor. It inhibits plant metabolism. It plays an important role in regulating abscission and dormancy. Abscisic acid function as stress hormone that help in plants to pass adverse environmental condition. Example, under severe drought, it prevents water loss by closing the stomata. It acts as antagonistic to gibberellins. Functions of abscisic acid: it induces dormancy of buds and seeds as opposed to gibberellin, which breaks dormancy. Involved with leaf and fruit abscission or fall, it inhibits seed germination and development. Abscisic acid also plays important role in controlling stomata opening and closing. Here is the summary of plant growth regulators, where produced or found in plant and major functions. Dear students, for each and every plant processes, phytohormones are involved. The role of plant growth regulators could be complementary or antagonistic. It could be individualistic or synergistic. The role of hormone is one kind of intrinsic control along with genomic control. Many extrinsic factors like temperature and light control vernalization, flowering, dormancy, seed germination, etc. via plant growth regulators. Dear students, thank you for watching. See you next class with another topic.